go into details, you compare your lane at 10 millimolar and the 250 millimolar. You can see the band there, and then why is the band in the 10 millimolar so more intense compared to a 250? Then another thing is it looked like the band at the 10 millimolar and the 250 they didn't show in the same size, because the size of the 250 seems like much smaller compared to the 10 millimolar. Can you explain this?
Yeah, because this is the first one that use the purification, because the last the imidazole you find last amount for the my protein in the nickel resin.
Okay, then why the
see, you can see here.
Yeah, we can see here.
Yes.
Yeah.
Yeah. This is the different size in the during the make the protein purification.
Yeah, can you go back to the Western blot just now?
Yeah.
Yeah, this one. Okay, I was thinking that you already got your protein, and how do you verify that this is your protein? Because when you compare to the post control, it seems like there's a lot of the massive band there, and compared to your purified 250 millimolar, and you put an arrow saying that this is your protein. How do you ensure to claim that this is your purified protein?
Victor, you can again.
How are you sure that this is your purified protein?
Because I'm using ladder. You can check the size for protein in with the control with the 10 imidazole, 20 imidazole.
Well, if the size is correct, and normally the post control is quite important because it can use as a comparison to your specific protein. But right now, your post control, we can see there's a lot of band there. Can you see the post control? Right?
Yeah, I can see.
Yeah. Normally, the post control, they must not, in general, it's supposed to show a same band compared to your 250 purified protein. But right now, your post control show like a series of band, and there's no any specific band compared to your samples. And how do you explain it in your thesis?
Yeah, Doctor, this is because after the make the 10 imidazole and the 250 imidazole, different the concentration for protein. You can see in SDS for the protein purification. Because the different size for band, because the different amount the protein during make the wash resin by the last concentration for imidazole.
All right. Okay. Can you go to the slide of the references, please? Okay, and I think that you put all right, the sentences are okay. All right, and the conclusion, please. Conclusion.
Yes.
Okay, last sentence. You said you claim that you already you concluded that you got something, and then it can be and proceed into the diagnostic platform already. Okay, from your recombinant proteins. From your study, can you conclude that what is the further step needed to be identified before turn your protein into a diagnostic application?
Yeah.
Okay.
Yeah.
What's the next step needed?
Yeah. Found the other method using the diagnosis for the Giardia, but I need the specific diagnosis for the Giardia. You can use it the MyProtein, make the tools. You can use it check the infection for the Giardia very fast, very fast, very fast, because the other diagnosis need more time, expensive, and something. But this is, you can make rapid test, use the direct diagnosis for the giardia, and the easy and the cheap the application diagnosis.
Okay. All right. Thank you so much, Ra'ad. Okay. Thank you for having a very good explanation. I'm done. Thank you.
Thank you, doctor. Thank you.
I'm done. So, who will be the next speaker? Yeah,
Okay.
Stana, right? So, thank you so much to Stan, and also to your sponsor, Dr. Sida. And we hope that to see you soon in your next presentation for during your Viva. Okay, good luck, Stan.
Okay, good luck, doctor. Thank you, doctor. Thank you.
All right, thank you. So the next presentation, are you ready?
Only one evaluator.
Yeah, I guess how many are evaluator? Let me check. Or maybe if there's anyone want to ask, Ra'ad also can raise your question here. If any lecturer or anyone who would like to have any questions. Let me check. One only, and also Cairo, sorry, Dr. Cairo also the examiner, Dr. Cairo. Ra'ad.
Okay, doctor. Okay.
Yeah, Dr. Cairo, please.
Oh, thank you, Heng. Thank you, Dr. Heng. Ra'ad, thank you very much for presentations. Okay.
Okay, doctor. Thank you.
Yeah, actually, I have a few questions, but it's already asked by Dr. Heng. Perhaps just want to add some more questions. For your expression, why you choose only 6 hours interval periods of expression? Why don't you go for longer, like 24 hours?
Doctor, because this is my protocol, I think in 6 hours. I discussed for this with my supervisor. Make the 6 hours.
6 hours. Do you think that 6 hours enough, or after you do the experiments and you did the Western blot, do you think 6 hours is enough for you to express the protein?
I think enough, because you can see my protein in Western blot in this time.
Because in order to expression is one of the experiments. Another one is you want to quantitate the proteins, right, and you want to use it for your diagnostic. So that's 6 hours enough for the protein to be used for your diagnostic purposes. If you look at your slide number 14, slide 14, this Western blot, okay? So I just wonder, at 250 millimolar if imidazoles, is this you purify the pellets or supernatant? Supernatants. Is this based on 6 hours or what? What?
5 hours.
5 hours. Why you choose 5 hours instead of 6 hours?
Because I'm during the expression for my protein, the successful in 4 hours. I'm making the 6 hours. You can check. You can use it the 5 hours, because already my protein make the expression during the 5 hours, between the 5 hours and 6 hours. You can use it between him in 5 hours.
Sorry, Dr. Khairul. Ra'ad, can you show the slide for Western blot, slide 12?
Okay, this, doctor?
Okay, yeah. And then explain now why you do 4 hours, 5 hours instead of.
Yeah. Dr. Khairul, see in C4 and C8, you can see the my protein expression during make the Western blot.
Yeah, I can see that.
Expression. you can choose this time because already in 5 hours and in 4 hours already my protein expression.
Yeah. Because you said that you are for your another purification, you choose 5 hours, the S7, right? But at your S8, 6 hours is also produced. If you say that your protein already produced, actually at 3 hours or 2 hours is already produced.
Yeah, my protein produced in 4 hours and 5 and 6 hours.
Look
You
at
can
your Western blot, 12/12.
Yeah, see. In 3 and 2, you can see is band into is very small. In 3 and 4 and 5.
S4. Okay, so if you look at your S4,
Yeah,
okay,
S4.
so which one the product that you want? Is that the upper one or the below one?
The other one is first one.
Yeah. Which one? The upper band or the below band?
The first band.
The
I don't
first
know.
ones, right?
Yeah.
So if you say that you are choosing 5 hours, which is S7, actually at S4 is 2 hours, it's already expressed.
Yeah.
Yeah.
I'm using the range between.
Okay, doesn't matter. Okay.
Mhm.
So because for your purification, it seems that at S7, at 5 hours, is very thin band. So I'm thinking, I have some thought that perhaps, is it possible that your protein has no optimal condition to be expressed during 5 hours, or perhaps the condition itself is not conducive for them to express the protein. That's why you got very low protein instead of a bulk protein. Any reasons perhaps you want to add? Any reason why you get very faint protein for 5 hours?
Doctor, you can see in Western blot, in 5 hours is that my protein in find in 3 hours find in 6 hours find.
It's very
I'm
faint.
choosing the
It's very faint. It's very faint.
Yeah, may I interrupt, Dr. Khairul? Okay,
Uh-uh.
now you cannot be so defensive when people ask you comment on your work. It's clear that on 4 hours the band is a bit faint rather than 5 and 6 hours. So it's actually Dr. Khairul. There's no reason behind this because Ra'ad he just did a very few work and then he's now in Iraq. So I think this now shows that you need to do more lab work improve your result. Okay, so
Yeah.
obviously you need to be here and you need to return here and do your lab work.
Yeah, because you have to show us the expression is really works. So if you produce this faint band, it seems like the expression condition is not conducive enough for you to express the protein. So if you can produce a bulk proteins, I think it's much better. So I think you need to extend the time hours for the expression. Yeah. Can we go for next question? Is on your preparations of the BL21 for the transformation process. So how you prepare your competent cells of BL21? So what is the method that you use for competent cells preparation?
you need the method.
Yeah. What method that you use to prepare to make sure that your cells is conducive enough to receive the foreign or vectors? What method that
Doctor,
you
you mean method for transformation
no,
the BL.
I'm asking you, what is the preparation for the BL21 you did to make sure that the transformation is work?
The BL21 using
the competent cell preparation. How you do the
okay. How do you do
competent
the
cell?
expression for BL21?
No, competent cells. You know what is competent cell?
Competent cell for the make the transformation, the making cell.
Yes. So how you do the competent cells?
I'm already my supervisor take me the BL21.
So
Make the
how you make sure that the BL21 has a very permeable membrane in order to receive the vector or plasmid?
Culture the BL21 overnight. Use the 1 mol for the BL21. Make the wash by CL and heating the in transfer my plasmid incubator
Okay.
1 hour.
Okay, thank you. I got the answer. So you are using calcium chloride methods, for your transformation, for your competent cell preparation.
Yeah.
Okay. So on Dr. Heng's question just now, you mentioned about these slides that you choose top 10 and also BL21. So you said that on top 10 there are promoters and BL21 has a promoters. So may I know what promoter that you mentioned on the host itself?
And BL21 have the 7 promoter.
What promoter that you mentioned? What promoter that you mean? What promoter?
The promoter for the RNA, DNA gene.
Can you explain more?
The doctor, the BL21 using make the expression because they have the ability successful for the expression because the BL21 have the 7 promoter. The other types
what promoter that you mentioned?
RNA and DNA.
Yeah, what is the name of the promoter?
7 promoter RNA.
Okay, I think sorry, Dr. Cairo, I just want to add out a little bit to you.
Yeah.
Dr. Cairo asking the name of the promoter. You keep mentioning is a 7 promoter. What is the name of that promoter?
Poly promoter.
The name of that promoter. Do you know that?
I don't know.
All right. Maybe you need to find out and write down your thesis. Thank you, Dr. Cairo. Thank you.
I think one more. For your Western blot.
Yeah.
Any Western blots, okay? So what is your primary antibody? What is your primary antibody?
I'm using the primary antibody for the histag.
Histag, okay. So for the secondary antibody?
I don't. I am already connect with the histag, the secondary and primary.
Okay. So perhaps last. So for your blue and white screening. So if you can look at your blue and white screening, okay, it seems like your blue and white screening, the populations of your bacteria is so condensed. So how actually you choose your positive positive bacteria, positive single population?
I'm choose white bacteria, good colony. I'm not choose the blue bacteria.
So are you sure that your plate has no contamination? Because
Yeah.
if I look at your
Because
plate, there are two populations there. There are big colonies and also single colony, very tiny colonies. Any reason that you have multiple size colonies?
no, I'm using this not contam. I'm using the same my colonies. I don't know why this where the difference between the size the colonies.
So how you spread your colonies? How you spread the culture?
I use I'm choose the active colony in during the transformation. After this, I'm already making the plate with the antibiotic kanamycin, making the 2.0 microliter by the XGAL, making the separate in the plate. After this, make the separate for my colony in the plate.
Okay.
Wait overnight. This is my result.
But I think if you want to use this picture again for your Viva, you have to come up with explanation why you got lots of multiple colony size instead of
Okay.
synchronized colony size, because I'm afraid that there are contamination. Okay, that's all from me. Thank you very much, Ra'ad. Okay, Dr. Heng.
All right, thank you so much, Dr. Khairul, for your very good question. And I think Raad kindly accept the good input from Dr. Khairul and then to improve your thesis. And I think that your sponsor, Dr. Shida, is a very well experienced researcher in this kind of the study, especially for the diagnostic application of the COVID-19. So try to improve your limitation in your work. As your sponsor mentioned that you are not away, and some of the experiment, it seems like you still not fully understood very well. So try to understand all the keyword and all the experimental step. Therefore, when you attend your Viva, you can answer all the questions given by the examiner. Okay, right?
Okay, thank you, doctor.
Okay. So is there any other questions from the floor? Any other questions? Any other lecturers got any questions to Raad? All right. If there's no question or want to ask later, and so I will shut the presentation for Raad here. Thank you so much, Raad, and hope to see you soon in Viva soon. Okay, good luck. Okay, thank you.
Thank you, doctor.
Hello, doctor.
Okay, the next presenter is Sana, right? Okay, Sana, my student. Okay,
Yes.
I will leave.
Hello, doctor.
Yeah, the next chairperson, please.
Tunggu Dr. Venu, kita akan start.
Dr. Venu or Dr. Eric?
Sudah, examiner Dr. Venu and Dr. Eric.
So if maybe anyone, Dr. Eric or Dr. Venu can be the chairperson or yeah.
Sorry, doctor, can you see my slides?
Yes.
Okay, thank you so much.
Okay, just wait.
I'm here already.
Just wait, Dr. Venu.
It's okay, I'm waiting. That's it.
Bob call ke Dr. Venu?
Dah. Saya dah WhatsApp dia kata kejap.
Oh, kejap, okay. So we wait for him lah kan. So kita nak continue dengan next presenter. Dr. Tashrik. Tashrik tak ada. Okay lah, we just wait a few more minutes.
Itu Dr. Zainul dah masuk dah tu.
Dia dah, okay. So we continue with next presenter. Now I would like to invite Sana, student under supervision Dr. Heng, and your examiners Dr. Eric and Dr. Venu. So you can start, Sana, if you're ready now.
Thank you so much, doctor. Thank you. Hello, doctors. Hello, students. I'm Sana Yasin, and I'm under the supervision of Dr. Leo Hen. My title of my research project is about characterization and immunoactivity of fermented selenous lentils, crude extract in macrophage cells. This is our final presentation. Sorry. This is my content. I will cover all the content here. First of all, it's my introduction and the background of my study. First of all, like my background of my study, since we are studying like selenous lentils is medicinal mushroom. It came from the idea like people right now looking for something for immune enhancing products. So, but this is came because like aging population right now and increasing by WHO, they related that information. Also, the second one, prevalence of the infectious diseases without an antimicrobial thing. So it made it significant global interest in food and medicines that put immunofunction, like not nutraceutical or immunoceutical things, or any agent like helping us with the immunology. So we went to the natural product. Okay, so natural product, one of the most common one, it's medicinal mushroom, came from the Chinese traditional medicine. So medicinal mushroom, it's like, for example, like the one that's most common, reishi, mfitek, and metek, have been recognized by the people and in scientific, field by their boosting property for centuries. So this is all came from their bioactive compound, like beta-glucans or terpenoids, play a crucial role in enhancing immune responses and reducing inflammation based on the experimental results. So, sorry. This is my study species, Selenous lentis, the wild one, and then it's common. It's known for it is biological active, anti-cancer, and immunomodulation. And the second thing, we are looking for the bioactive compounds inside the Selenous lentis. The most common one and the one like enhancing the biological activity is polysaccharidine and terpenoid and phenol. And for example, why we are looking for the bioactive compounds? Because like inside each of the class of bioactive compounds, there is like active compounds or key active compounds under this class of the compounds. For example, here we have histidine and inoculon, one of the highest and strongest antioxidant activity of the phenols. They found it from Selenous lentis wild one. So what's the benefit of finding this one? So it's a benefit for the application in natural immunomodulatory agents there's a new field. It's called immunoceutical. Like people can get it without any side effect and toxicity. And the second one, potential for clinical application. For example, like in its application, for example, like in liver toxicity, any kind of the application. And third one, for drug development, fundamental units. So the focus of my study is about immunoactivity of Phelinus lentis, the fermented one. So I did a literature review about how it's going on. So the first one, humoral and cell-mediated immunity with the adaptive one, and we have innate immunity. My focus is going to be innate immunity, but I just like studied all of them. So first of all, we have T cells. Like the effect of the Phelinus lentis extract on the T cells, enhancing cytokine production, interferon gamma and interleukin-4, and Th1, Th2 balance the cells, and also lymphocyte proliferation, like splenocytes. Most of the studies have been done in vivo in the mice. So the second one, B cell, also like its effect on the B cells by the proliferation of the B cell, and also augment antibody production. So two main of the function of the B cells. And the other side, it's macrophage. For the macrophage cells, it's affected like by macrophage activation and polarization. We know like macrophage after the activation is going to be M1 and M2 classical, and the other one, alternative one. And then we have phagocytic activity of the Phelinus. It's enhancing by the polysaccharide or any compounds like extracted from Phelinus lentis. And the other one, nitric oxide production, also is one of them. Responses by the macrophage, like for the any inverted like microbes or any kind of the immune system facing the like fight, like any kind of the responses. So on the other one, cytokine production. So I mentioned like my sample is going to be fermented Phelinus lentis. So my sample is going to be a new type of the fermentation innovation of the fungal processing pathway. This idea came from after like Phelinus lentis. It grows on mulberry tree, and it's so hard to collect that mushroom, and also like it is grown like in between 2-3 years, and the fruiting body of that mushroom, it's hardly people can get it. Most of the products in the like the markets and everywhere, like medicinal market, the common one, like Chinese medicinal market, most of them it came from the fermentation or processing, like downstream processing by the submerged fermentation. The products all produced by the mycelial growth, not the fruiting body, not the whole one. So the newest fermentation type, they call it, there's like two of the new ones, submerged fermentation and the solid state fermentation. Submerged one, I'm not like going down to it because it's not like my focus, but I just want to explain that part. Submerged one, it needs water, and the mycelia will grow and release all the metabolite inside the water, and you have to feed the water. But the other one, solid state fermentation, is the same as how the naturally growing the mushrooms on the like that tree, mulberry tree, or some places. So that one, solid state fermentation, they are using substrates like brown rice and so many of the substrates, but the brown rice is the most common one. They will put the mushroom with it, and then the mushroom will grow with the substrate. And then this is like the idea. It's the cultivations and processing of fungi, addressing the community needs and expanding its application, because we are putting substrate. So it means like as the hypothesis, it's gonna be enhancing the releasing the metabolites more. So there is they have done like 3 studies on the solid state fermented Phelinus lentis extract effect on the vital state. The first one is showed like anti-inflammatory activity, and the other one anti-inflammatory effect, and the other one anti-inflammatory. Both of them anti-inflammatory, but in different one. The one like on the macrophage cell, and the other one, the third one, is on the animal-based in vivo one. So they have done like in 2009, 2010, 2014, and from that time, there is no other study, and also there is no other study about the characterization and what's like, what is the active compounds and what's the characterization of the crude polysaccharide. So problem statement of my study is going to be the immunoactivity of fermented Phelinus lentis extract in macrophage cell line with the determinate extract or crude extract because it lets the research to provide scientific background of the effect of fermentation on bioactive compounds. So my research question and hypothesis, the first one, how like fermented polynyl planters, then by hot water extraction, influence the antioxidant activity and nitrous oxide production and the transcriptional profile of the pro-inflammatory cytokine production in the treated or differentiated THP1 cells. As we said, like this one is fermented, there's like no evidence, there's no background, scientific background about the outcome, how like the fermented one, is it working the same as the wild one? So the hypothesis of this study, the polysaccharide from this one, the whole fully fermented polynyl planters, it's gonna be as wild type, will significantly modulate human macrophage cell line, THP1 immunoresponse. Also, like most of the study have been done on the murine macrophage, not the THP1, not the human monocyte. So this is, I have 5 specific objectives. The first one, characterization of the bioactive compound. Second one, determining the antioxidant activity of the crude polysaccharide, and then determine the extract from third, fourth, and 5, it depends on the cell culture, determines the cytotoxicity and cell proliferative effect on the differentiated THP cell, and also investigate the pro-inflammatory cytokine gene expression on THP1 cell after treating with the polysaccharide, and evaluating the nitric oxide NO production in the treated macrophage cells. My methodology, the main methodology is depending on the hot water extraction, the one like naturally have been used back of the day. This is my mushroom. I got it like powder because mine it was polystate fermented, but I just put it in the picture. So fine powdered fermented polynyl planters, and then by hot water extraction, the ratio 1:14 for 4 hours, like at 80 degrees Celsius. And then purification by the 95% of the ethanol purification. Yeah, the simplest pathway with the extraction, because this one, people also like outside of the lab, they can use it. Okay, then air dried crude polysaccharide, crude polysaccharide, then go under the, experiment. The first one, I have done my, the first method, it's small system identified from my polynyl planters, to be sure like the sample, the species they have used it is polynyl planters, because as the morphology of the polynyl planters, most of the polynyl planters, and there is a there's another species, and under the different units, it will look like the same. So it needs the moisture system identification to make sure like your sample is a polynyl planter. And then phytochemical characterization due to the colorimetric assay. I have done like polysaccharide, beta-glucans, tretinoin, total-phenol, total-flavonoid in triple sample, duplicate, and then antioxidant activity, the free type of the antioxidant activity test, DTPS, FRIP, FRAT, and ABCS. And the last part of my research, it's my methodology is based on the cell culture. We are like dealing with the THP1 cells in the lab, and also like goes under the MTS assay, natural oxide production, and then after the treatment, we have to do the gene expression by real-time feedback. This is my research flow chart. Crude polysaccharide extraction, the main part of my job is I have done the characterization of my crude polysaccharide, and then goes with the in vitro immunoassay, THP1 cell line. My results and discussion. First of all, my study species sample DNA identification. I visit ITS 4 and number 4 and number 1 for my primer to identify the strength of my sample, polynyl planters, and the other species, like I use it as a product control. After I got my band, after the PCR product, and I use the ITS 1, ITS 4 product control. Also, like why I use it, there's two jealous for it, because the first time I didn't get my band for my polynyl planters, and then we tried again, we got it, and also like we use it different primer, but I will explain that thing. So, but at the end, I got it my results for my cleaner census with the ITS1 and ITS4, not the new payment. That's why I didn't put my data here. So then after the DNA sequencing, we send it for sequencing. The data, the raw data, we got the raw data, and then I did the blasting for my data, and then the ID score covering, then finding the with the highest score, and then I did the whole system, phylogenetic ID identification for fungi. This is my result. So the first one, example cleaner census belongs to that point, and this is the NCBI ID, gene bank information, and the similarity score was 98, and the E value was 0, and the rest was I used it as a focus control from IPS region. So the genus Rhinos, Grifola, Sweden, and I'm not familiar with reading their name, Pleurotus irrigini. Okay, first of my objectives that I covered in my work, characterization of pure polysaccharide, the bioactive component. I did the total carbohydrate content, total phenolic content, total sericinoid content, and total flavonoid content. I did the hot data extraction. My project is all about hot data extraction. After I got my results, the total carbohydrate is 132.8. It was high, and the second one I got total phenolic. It was not that much high, not that much like low, because when comparing with the wild one or the other species of the medicinal mushroom, it was like low. And total tritipsine 1.19, and I didn't get total flavonoid, because I didn't get total flavonoid, and there is a paper saying like there's a total flavonoid in the wild one. So I did another tried another method for my confirmation and comparing. So I did the ethanol extraction. So I got as the same. I got no total flavonoid. So then I just stayed with this result. So in my sample I don't have total flavonoid. But the good thing, I got total, total certified hair with ethanol. But for the total polysaccharide, because my focus is going to be polysaccharide, or because amino activity belongs to the polysaccharide or the beta glucan. So that one with the hot water, it was threefold higher. This is a diagram of my comparison. Only the certified was higher in the ethanol extraction. This is due to the structure and molecular structure of the organic of the certified. The second one, after I got my polysaccharide, the higher level, so I did the glucan content. The main component in the total carbohydrate is glucan that have two types, alpha and beta. So I did the experiment for them and contribute to the studies, the immunomodulatory effect and antioxidant effect mostly come with the beta glucan. So this is my result of the beta glucan. Total one, it was 56, more than the standard kit, and also only I got a little high level of alpha glucan, and also the beta glucan is 36 percentage. This is the diagram of my glucan content, and here is comparison between beta glucan percentage in the control beta glucan and my fermented selenium census. So it was significantly different between them. Determine the antioxidant activity. My second objective, determine the antioxidant activity of fermented selenium census in polysaccharide. So here is my IC50 values of mg per ml of ascorbic acid, fermented selenium census due to the radical scavenging activity. The first one, I didn't get IC50 due to the low amount of the compound responsible for antioxidant activity. Only the froth gives the result for the IC50. Here's the diagram of the froth. So it was more than 50%. It can do the reducing activity, reducing ability for the froth. The antioxidant inside my sample, it could reduce the the froth, reducing Fe3 plus to Fe plus 2. So the result, it shows like the less potent antioxidant activity of sample compared to the standard, but it's still like there is the antioxidant activity. So antioxidant activity for the DTPH and also FDS. So I didn't get the IC15, but also like their activity, it was like 34.9 for DTPH and also 47% inhibition in the FDS. While this value don't allow for the determination of the IC15, but also like indicate that there is extractively scavenging activity, small portion for both of the DTPH and FDS radicals at the tested concentration that I used 10 mg per m, my highest dilation sample. To conclude my characterization and antioxidant activity results, first of all, like the extraction, when you are dealing with the extraction is significantly the method that we are using affect the composition and yield, and what the extraction yields higher total carbohydrate, because like I'm focusing on the polysaccharide, so for me it was better, but for terpenoid, it was like ethanol extraction was the yield better amount, and the polysaccharide, the one that it's giving you the result, the higher one, it's better to be characterized by structure composed. Characterize it, and the structure composed of beta glycosidic linkage. It means like this type of linkage. It gives the it's related to the immunomodulation and antioxidant activity of that compound. Impact of the phenolic content and antioxidant activity. After like I compared my results with the other papers, so my phenolic, it wasn't that much low, but didn't give them because the amount of the phenolic in mushroom extract directly influences their radical scavenging. So why mine it wasn't? Then I forgot to cite this one. Then the antioxidant activity, sometimes it's not depend to the amount of the total phenolic. It depends to the specific compound. For example, inositol, one of the strongest antioxidant activity in the Philina synthesis. So flavonoid content in the wild Philina synthesis in Thailand, they have done the research. So they found that there is no, the only species of Philina genus, Philina synthesis doesn't have flavonoid. The characterization result is variable among the studies that have been done because of the temperature extraction methods and ratio of the dry powder to the solvent and type of the solvent. This made my result interpretation hard to compare with the other paper, because each of the papers was everything was variable and everything was different, so it wasn't that much like giving the significant like explaining. But and this is my reference, and this is thank you so much. Just let me to go back to my slide, because my method is contains cell culture. So, but my working is just stay here, because since I started, I'm just like working on my characterization and working with the cells. Right now, our cells like we didn't proceed with our cells like to treating it because of the instability of the cell growth and low growth rate of our cells, and struggling with the cell culturing. So we are figuring out our cell culture. That's why I don't have any data about cell culturing. Thank you so much for your listening.
Thank you so much, Sana, for your good work. I believe we have done a lot of work and nice presentation, a nice slide, comprehensive presentation. So I would like to congratulate you on completing everything, and congratulations to your supervisor too, Dr. Herb, for supervising good student and good project.
Thank you so much.
Okay, now let's go for Q&A session. Now I would like to invite Dr. Eric and Dr. Venu for any questions to Sana.
Dr. Eric first, can?
Yeah.
Okay, thank you. Can you go back to objective? Can I see the objective again?
Yes, sir.
So you walk.
Yes.
So your
Yes,
work is complete already, or what?
No, doctor. Like First of all, I didn't know like our final presentation is gonna be in November, because we were like discussing with my supervisor. Our work with the the other three objectives is gonna be on November, but then we just like knew that thing, so we just present the result that I got it, because mostly like I was busy with my characterization. I mean, I did two methods like to just like get my result done.
So the experiment you carry out is you done the THP1, the macrophage cell assay, the cell.
No,
You
that's
did
it.
that? Oh, why?
Only growing, because we are like teamwork, like we are working in team. So
Oh,
with
okay,
my
okay.
seniors, we are like dealing with the cells, like kidding and packaging. But I don't know if you are aware of that thing. Like sometimes nowadays, like we are facing some of the low growth and instability of the cell growth. This is the problem that we are facing right now.
Okay, okay. Because how many month or how many day you left before you have to submit the final thesis?
I think that's like less than month. This is just less than month.
December.
The 8th of December. Like I don't know, like it's like short time, and also like as a mixed mode.
Maybe 2 months left. Maybe about 1 to 2 months left, I think.
Uh,
So I think
yes.
maybe my advice actually, if you cannot complete some of the experiment here, maybe you should think just cut off the objective, because you have 5 objective here, It's
Yes, that's it.
consider quite a lot for the mixed mode student also. Maybe you can cut to 3. My suggestion this you have to discuss with your supervisor,
Okay,
because
that's
no point. I think you don't have time. If you don't have time to finish up the experiment, better you just reduce the objective of this. Maybe just reduce to 3. The fourth, I think, because if we go back to slide number 17,
yes, that's it.
17, 17.
Okay, that's it. Yes, that's it.
Here, you see, because here is a ascorbic acid is a control, right? It's a control, right?
Yes, that's it.
And one thing you notice or not, you want to do the antioxidant DPPH, you don't get the reading. Okay,
Yes,
ABTS,
yes,
you only get the reading of FRAP actually. Is this
yes.
good FRAP readings compared to the control?
No, that's it. No, that's it.
So that's my question, because if this is not, because usually standard for oxidant test is DPPH that's the most common people use for antioxidant, right? That's common
Yes.
actually, that's a common. Why you didn't get the result for this one? No result, or is unable
No,
to determine, or is no result?
unable. No, doctor, unable to determine. Even here,
Unable to determine. Okay.
let me to show you I have my table on that one. I just like put here, the DTPH, it was like 34.7, because I only did for hot water extraction. So maybe due to the low amount of the phenolic,
Maybe
because
you should
phenolic
put
and
this
black
one,
light.
You should
Oh,
put this one instead of the not able to determine, because at least this has got some result. Look
I
back
think
on you, because remember your test sample is on the this one, the fermented linters, not the ascorbic acid. Is look back on the thing, how you present the things. It's
yes,
better you got result, but maybe it's not as good as one, but it's better to put the last slide that you show. At least you show some result on it. If not, it's just blank, right? It's just everything's blank, nothing to show. Actually, look bad on you,
I just
because
like
your DPPH is not one to show on the ascorbic acid, because that's standard vitamin C, right?
yes.
Nothing to tell people to do it, but your one is actually, I don't know it's good or bad, because you want to show you got activity is good, or you don't want to show activity is good. I don't know. It's based on the interpretation, but better don't show the ND. Not able to determine. It's not good on your things. You should show you've done some result, but it's the result maybe is not living to the standard or this kind of thing. At least show, but not put on the ND, because remember this is your experiment.
Yes.
We are not doing ascorbic acid. Is you do the fermented one actually. At least you put some result, not ND. At least.
Yeah.
So
Thank you so much,
next,
doctor.
next slide.
Okay. I put it here. I don't know because
this
I
one.
didn't want to.
This one same also. I think you better discuss. I think
Yes, I
now
discussed it
actually,
here.
if you can finish out the other experiment you do, yes. If not, I think better just cut off. We write or do some more discussion because
Mm.
I don't think you can finish the cell one. I'm not sure. Are you doing currently the cell line already? No.
No, just like observing. Like just
See,
passaging the cell, but the growth is too low. The
but if
confluence
you
is not.
you can do, you finish it lah. If not, better you discuss with supervisor. Maybe just limited to your objective, so you don't do bad because you cannot complete. No point. When you submit the thesis, you need to complete the thesis.
Yes,
I think you only have maybe 1 month only left. I think December you have to submit already. I'm not
yes,
mistaken. December, right? Early
yes.
December or
The
mid
first
December.
the first week of December.
You see, you only have 1 month left only.
Yeah, yeah.
So don't be too ambitious lah. I think because people understand you're a mixed school student. Discuss with supervisor. Dr. Heng should be understand this kind of problem. I think
Yeah,
discuss the thing. Not put ND because ND look like you don't do anything at all. Put the experiment is some result is okay. Don't put ND. ND is look bad on you. And discuss the thing.
Okay.
If can do, can finish. If cannot, because on December you have to submit the final copy already. You don't have time. You only maybe you need write. You start writing already. I hope you writing part of the. Yeah, I hope
Yeah.
you writing the introduction, the methodology, the research. If not, try to have it lah. Okay.
Yes, thank
That's
you
all
so
my
much.
question. Because you put too many objective after that, finish already. Then after that, I don't see
Thank you
anything.
so much, doctor.
Okay. That's all my question. Thank you, Chairman.
Thank
Chairperson.
you.
Thank you, Dr. Eric. Okay, now Dr. Venu, you have interested.
Yeah. Hi, Sana.
Hello, doctor. Hi.
Yeah. Your presentation is good, like what Dr. Shaida said, but I had the same query as what Dr. Eric have mentioned.
Yes.
Okay, you have so much of objective, but certain objective you couldn't complete.
Yes.
Okay,
Doctor.
so the objective that you can't complete, you must omit it and just show that whatever that you have completed. Okay, so like
Okay, doctor.
showing ND data, so not determined is not good, like what Dr. Eric said. It's not good.
Yeah, thank you so
And
much because this is my first, research presentation. Maybe that's why I didn't know that thing. Thank you so much.
Yeah,
It's
yeah.
so good for me. Yeah, this is a good point.
Yeah.
Thank you.
So when you have 3 objectives, you have completed. I think the first 3 objectives you have completed, right?
Yeah, most only the one like related with the cell, but the problem is just like things going on. It's an unexpected one. That's why, and I did 2 methods for confirming my results thing too. Busy with the like characterization. That's why. Sorry, let me go back to my. Here's my objective.
Yeah.
2 of them is finished. Only like the third one is depending on the cell.
So I think so your 4th objective you have not done anything yet.
No, because of the cell condition.
Yeah. So I think you can just complete your 1, 2, 3. Your 3rd objective you complete it, and then you just write down and remove 4th and 5th objective lah. Because if you don't complete, you put, then there's no data for it, and it is indicating that you have didn't done your work.
I see. I will discuss that with
Yeah.
my supervisor then.
Yeah,
Thank
yeah.
you so much, doctor.
You need to talk. Yeah, you need to talk with Dr. Heng. I think Dr. Heng also understands that.
Yeah, of course.
So yeah, so you don't have to worry because you don't have to finish 5 objective. You are just mixed mode. Okay, everyone, even the examiner understands that. But when you submit your thesis, there must be some data to show that you have completed the objective that it is been mentioned there.
Understood. Okay, thank you.
Okay. Yeah, I had the same when I was looking through. I had the same thing with Dr. Eric. So
yeah.
that is the main part that you have to get it done. and finish your objective 3, at least your cytotoxicity work.
Okay, I see. Thank you so much, Dr. Benyo. Thank you so much.
Yeah.
Ah.
That's all for me, Dr. Shaida.
Sana,
Yes,
Sana, I
Doctor.
want. Yeah, can you go back to the next slide?
Next
Slide
slide,
with
Doctor.
the
This one?
ethanol and the hot water extraction.
Okay, Doctor.
Okay.
This one, Doctor.
This one, yes. This is I think it's the carbohydrate component is higher, right? They have how many time? At least four
Three
times higher than the ethanol extract. Yeah lah,
Yes,
but
okay.
the tripidins is almost lower lah compared to, at least it's got extraction on the. So this one I think you should highlight the way how the result different, you know, and what the purpose what you use, because you use hot water, right, and ethanol, this kind of extract. I think you should, when you do a discussion, you should highlight what the purpose of this two different type of method of extraction, hot water or ethanol. Let's say you want a different type of content inside the extraction, right? So let's say you want the tripidins, three terpenes in higher 3 times lah. It's nothing lah, can say, but it's at least you can highlight the differences between. The carbohydrate is what? Is glucose? Nothing,
Yes,
right?
Doctor.
Phenolics,
Yes.
nothing.
It's polysaccharide.
Glucose, nothing, right? It's not affected on the phenolics also. So in the next biotic compound, people usually don't discuss about carbohydrate for extraction, right? For compound, right? That's usually you have to usually people will leave phenolics or this kind of things. This is got better result, double or twofold compared to the ethanol extractions. I don't know. Are you compare the other experiment? Are you compare the hot water and ethanol extract in the next experiment?
No, can I just explain this one? The ethanol extraction, I have done this one for myself and for my literature, because like in the literature, when you are like doing writing, they are saying like there is a phenol, like there is flavonoid, there is these trichinoids. But when you are doing in hot water extraction, maybe you are not getting things. So that's why when I didn't have my flavonoid, so I just wanted to make sure like I will have my flavonoid by the ethanol extraction. So just like this is that part, like by ethanol, the solvent organic one, so I can get my flavonoid, and there's flavonoid that cannot be detected by the hot water extraction. So I did this part only for confirming my results and make sure like about some of the papers, but like some of the papers like mentioning something, the results saying something, but when you are like running it, the results not the same. So one of the problem of the that's why like Dr. Herring he chose this project, because there is no standardized results and calculation and the background of the characterization. Whenever you are doing, when I'm comparing my results to a paper thing, so someone thinks it's not matching. So for example, this one, yeah. Thank you.
Okay, I understand. I understand, but you need to highlight the because like I say like not much result inside your finding as the moment. You need to highlight for discussion as the thing. I'm not sure lah. Maybe you can compare with the other researcher data or this kind of thing just to highlight any differences. If not, very difficult lah. I know it's quite difficult to write on the discussion on other things. If not, how are you gonna present it because you don't have really much data. Anyway, that's all my question. Good luck, Sana.
Thank you so much, Doctor. Thank you so much.
All right, thank you so much, Dr. Eric and Dr. Venu, and thank you, Sana, for your presentation, and all the best to you.
Thank you so much, Doctor.
Yeah, okay. So I pass the session to next chairperson. Apa?
Yeah, I'm here.
Oh yes, Dr. All right, thank you.
Thanks, Dr. Shaida. All right. I checked the list. Qingdao is around, and then Dr. Tashri, are you around?
Yes, I'm coming.
Okay, great. Thank you. And then Dr. Eugene. Dr. Eugene.
I think we need to wait a minute for Dr. Eugene. She knows already.
Okay. Yes. So we wait for Dr. Eugene. Thank you, Dr. Chong.
Maybe Jintao, you can share your slides now. Dr. Eugene said he's waiting for WebEx to load. So I think he'll be joining us in 1 minute.
Okay, great.
Hi, good morning. I'm joining by phone because I do not know why my PC cannot join in. I'm okay now. Thanks.
Okay, thanks, Dr. Eugene. Okay, so everybody is here. So the third presenter will be Jing Tao Pan, student supervised by Associate Professor Dr. Chung Yi Siu. Jing Tao, you may start now. Please go ahead.
Morning, everyone. My name is Jin Tao Pan, and my supervisor is Dr. Chong. And my topic is pentapeptide identification for the inhibition between caligulin B S189 proteins by in silico approaches. It's my context. In introduction, from 2060 to 2033, there are three 370 new drugs was approved by FDA. The number of peptides and oligos occupied almost 10% of new drugs. Despite the rise of small molecule drugs in 7 years, peptides still occupy an important position. Short peptides are cost-effective products with high purity and more bio-friendly as bicycle biology molecules. However, they have some limitation, such as short half-life and low overall bioavailability. And we have response methods such as terminal protection and using unnatural amino acids. So there are so many peptides used in skincare and some ocular disease, such as CMV and dry eye. And in the mouse eyes, the intravitreal half-life of peptides is known to be in the range of a few hours. So the peptide drugs is important, but how can we design or screen the treatment peptides? There are two ways to build peptide libraries. First is the chemical method, SPPS, involves sequentially adding protected amino acids to end of a growing peptide chain, ushered to an insoluble resin with raising steps to remove excess regions and deprotect the peptide chains. Second is biology methods, to produce DNA library first and then putting them into phage for merging with phage's DNA, coding co-proteins. Those co-proteins contains our potential peptides, so we can easily using targeting receptors to select our treatment peptides. Why targeting S109? But those methods rely on random selection and extensive preparation, and subsequent preservation level must be expanded. So virtual screening wherein cyclical approaches demonstrated that they are less difficult to use, process a high throughput screening, and are economically efficient in peptide development for drugs. Apart from that, the result of virtual screening offering 3D interaction information. So why we choose the S100A9 for targeting? S100A9 monomers looks like M shape and is easily assembly to amylose, not only self-assembly but also assembly with other members of S100 family. And the S100A9 amylose cause lots of disease, retinal toxicity, AD, and cancers. Amylose structure of S100A9 is involved in regional dysfunction, and S100A protein as a molecular target in the ocular surface inflammation disease. And is the S100A9 amylose is the attachment core of A beta fiber for progress of AD. They have different kinds of the structure, can be hydrophobic, templating, coating, and losses interaction with A beta fibers. And this picture shows the S100A9 on the surface of A beta fibers. Next is how to screen a treatment peptide. So there are 3 S100A9 inhibitors can block amylo aggregation of S100A9. ApoE appears to be used to clap S100A9 homodimer in solution and restrict its extension. And this article mentions from less than 50 to less than 54 is the connect area between both ends of each S189 homodimer in S100 fibers. It's worth mentioning that only oleate, a natural killer chemical in olive oil, has the ability to disaggregate S189 fibers about the pivot stick between tryptophan 88 and hydrogen bonds with adenine 85 and glutamate 52 of S189. Consider the cost of peptide production and the number of screening. The number of peptides increase 20-fold when each amino acid is added. So we use pentapeptide library to screening the treatment peptide, which can targeting those active area already be published. Hopefully one day after validation, the pentapeptide treatability, the pentapeptide drugs can load it on contact license for long-term treatment, and maybe it can use to treat the AD disease delivered from eyes. Is my program statement. S100 self-assembly is relevant with many diseases, and to block S100 self-assembly is required to identify novel peptide drugs that are more biological friendly and have stronger binding ability. And build a virtual peptide library makes it simple to uncover prospective medication for treating not only S100 assembly, but also additional disorders in future. Research aims and objective. First, to construct a pentapeptide library for virtual screening. Second, to identify the pentapeptides that can bind with S100. Third, to evaluate the interaction between pentapeptides and S100. It's my methodology. Some 3D structure of molecules will be downloaded from the protein bank and ChemiSpider database. OLA will be my positive control to compare the binding ability of peptides. Producing the pentapeptide library via RDKit, a Python API, it can transform the peptide sequence to PDB format files. And the OpenBaba will be used in converting the PDB to PDBQT, because the PDBQT is the spatial format used in Autodoc. Binding free energy and hydrogen bond of Autodoc results will be analyzed and ranked. The most detailed analysis will be used the AMBER 2080 to do molecular dynamic simulation. And the Python will use in batch process and manage the data. They are my results and discussion. In recent research, I use arrangement and combine of 20 animal acids in 2 sites, resulting to 400 results, and then putting codes into the 400 results. We can produce any peptide library when change the codes putting in the middle. For example, we put the same 40 results into the middle of the 400 results, can generate the tetrapeptide library, including the 160,000 peptides. And the pentapeptide library can be achieved by putting the 20 animal acids into middle of each peptide in tertiary peptide library. 20 multiply 160,000 equal to 3,200,000. And using RDKit API 10 days to build the pentapeptide library. The size of PDB library is 28.7 GB. And then we convert to PDBQT format. Next is we use Autodoc to docking 300 times between OLA with S100 to reproduce the docking results. And the binding free energy is negative 8.23 kcal per mole. our result is similar with the results of articles. And then we meet a big problem. We don't have enough resource to docking all pentapeptide library. So we docking the tripeptide library with S189 first, because the tripeptide library only 8,000 peptides, and then choose the tripeptides with high score of binding free energy as code to isolate the pentapeptides. Using those pentapeptides docking with S189 to screen the potential treatment peptides. Is the docking results of tripeptide library? Three tripeptides exhibition binding free energy at negative a 0.0 kcal per mole, which is close to ODA's binding free energy. So we choose the three tripeptide as code to isolate the pentapeptide library. Because the pentapeptides with WWF and WPW codes are more likely to provide a large number of outcomes with lower binding free energy. Especially those pentapeptides include the WPW code. There are two docking results lower than negative 9 kcal per mole. The WPWTY with the best binding free energy binds outside of S189's active site, so we incorporate the WWPWH with the second best binding free energy into the MD simulation process. And the other one, the WPWYW peptide binding. Sorry, Sena, can you?
Sana, please switch off your mic. Please unmute. Sana. Thank you. Okay, sorry for that. Jingtao, please continue.
The WPWY peptide with binding free energy negative 0.7 also was chosen because it has the greatest number of hydrogen bonds with S189. There are 7. So we successfully identified 2 pentapeptides with potential to inhibit S189 assembly. Their binding free energy is lower than ODA and have more interaction with S189. And then we saw 100 nanoseconds MD simulation 3 times to discover more details of their interaction with S189. It's the MSD data. 2 peptides binding more strongly to S189 than ODA, because the MSD represents the distance between ligand and the receptor. If the ligands move too far apart, they may break away from the receptor. And the MSD curves from 3 times MD simulation of ODA and S189 are unstable. You can see the black lines, they are unstable. Next is the RMSF. RMSF measures the average atomic mobility during molecular dynamic simulation. In other words, the lower value in this residual site means this residual more stable during MD simulation. And expect the results of the second MD simulation, the WWPWH peptide shows a more stable binding ability with S189 than the WPWYW peptide. And the ODA only in the third time MD simulation showed the better MSF results than both peptides. However, MSF data shows like 3 compounds have a similar density to bind to S100A9. This data shows the OLA generates less hydrogen bonds with S100A9 than the two peptides. You can see the number of hydrogen bonds of OLA can create with S100A9 is less than 6. And where WWPWH may generate up to 11 hydrogen bonds with S100A9 on occasion. Is the number of the hydrogen bonds between S100A9 residues and ligands during the 3 times MD simulation, and the hydrogen bonds number is less than 1,000 were not shown. And the y-axis is the exponential axis. You can see it's the 100 and 1,000 and 10,000 and 100,000. So compared with that of ODA, both pentapeptides have wide range of the hydrogen bond interaction with S100A9. From glutamic 52 to aspartic 65, and lysine 93 to proline 107. And this is MMGBS results. It's a pro-minor approach for determining the binding free energy of ligands to proteins. Binding free energy results suggest that two peptides combine stronger with S100A9 than ODA. During MD simulation, ODA's total binding free energy was 5% greater and near 600 kcal per mole than the two peptides, and the difference is highly statistically significant. Then we focus on the contribution of individual residues. The hotspot residues contribute significantly to the total binding free energy of the complex, typically lower than negative 1.0 kcal per mole. In comparison to ODA, the binding free energy of two peptides not only is contributed by the potential active sites from agonine 55 to alanine 89, but also by other active sites from leucine 49 to valine 58 reported in some articles. And this the data indicates that the two peptides can have additional interaction based on ODA's interaction with S100A9. And the data also overlap with number of hydrogen bonds in S100A9 residual results. 52 to 65 and 93 to 107. Okay, is the conclusion. And we successfully constructed the entire pentapeptide library, and it was released on USM library, and then screened the possible therapy pentapeptides and assess their binding ability with S100A9, which is superior to the natural products OLA. And in comparison to prevention inhibitors, the two peptides will interact with S100A9 residues from adenine 85 to alanine 89 and leucine 49 to valine 58, indicating the significant therapeutic potential. And recommendation for further research, the two pentapeptides should be evaluated and verified in one experiment to demonstrate the potential to degrade S100 fibers while also inhibiting the elongation of exciting S100 fibers. And then we can use some animal models with an AMD will be employed for in vivo invalidation of two peptides like amino can solve the regional toxicity caused by S100 fibers. Okay, this is my final presentation, my flowchart and Gantt chart and reference. That's all. Thank you for your time.
Okay, thank you, Ching Pao. I would like to invite Dr. Ashrik to ask questions or and give the comments. Dr. Ashrik.
Okay, thank you, Prof. Ong. Okay, hi, Jing Tao Pan. First, I would like congratulations for finish your research work. Okay, I think that's a hard work lah. All right, I have few question. Okay, if we look back on your page 32, okay, if you go back for your page 32, okay, and I think also page 31 of, yeah, from 31 until 32 lah. So you talk about hydrogen bonds in these interactions, okay, between the peptides and also the calnogorine. Okay, my question, why you only choose the hydrogen bond for the analysis, and why not the others bond like hydrophobic, like maybe salt bridge or ionic interaction, so on? Why only hydrogen bonds in your study?
About this question, in these articles about the inhibition of the S100 amylose, they only mentioned the peptide staking and hydrogen bonds interaction with S100. And the second is hydrogen bonds is an easy interaction we can analyze. Yeah.
Okay, meaning the other interaction like hydrophobic we cannot analyze using this any software because you have the all atomic trajectory, right? So
yeah, but actually they including all of the interaction such as in this the binding free energy resources. Yeah, so like one vita and something else. It can analyze all of the interaction, but for easy to show to others, I think the hydrogen bond is we can directly see what happened during the interaction.
Okay, all right, and then okay, so you're using MMGBSA, right? Molecular mechanic generalized bond, MMGBSA surface area lah. Okay, so can you mention me others than MMGBSA that usually the researcher use, other than MMGBSA? What else that researcher use? Other than MMGBSA, I think we have another one more. If protein-protein interaction, other than MMGBSA, we have one more.
What do you mean? I don't get your point.
Okay. In your study, you're using MMGBSA, molecular mechanic with generalized bond and surface area. But other than this analysis, what else that usually the researcher use for the protein-protein interaction analysis?
Actually, they only consider the binding free energy actually.
Yeah. Binding free energy also can be used by this analyzer MMGBSA, and then we have another thing, another analyzer MMPBSA, not G, not B, but P. Poison
Oh,
boxman.
I know.
Okay.
Uh,
Okay, poison boxman. So maybe you can try to find the difference lah after this. Why you using GBSA and why not PBSA and what the different and what is others lah. Okay.
yeah,
All
yeah.
right.
I think different is about a solution, and I can remember all of the things I read it
Yeah.
before. Yeah.
Yeah, okay. But it's not but actually if you have the trajectory of your MD simulation, you also can analyze by MMPBSA. But
Yeah.
MMPBSA, I think it need a huge power of computer in order
Yeah.
to get the value. But actually this analysis more suitable lah for the protein-protein interaction because your
Yeah.
peptide is protein. Okay, usually MMGBSA we're using for the protein ligand, but it also can be used by for protein-protein interaction, no problem. All right, I think that's all first from me. Maybe we can pass to Dr. Eugene for the next question. Prof.
Okay, thank you, Dr. Tashri. Dr. Eugene? Yes, thanks for
yeah.
the call, Dr. Tashri. Yeah, so maybe I'll just start with a simple question. In your conclusion, you mentioned something about the USM library. Can you explain a little bit more about that? Release on USM library. So what does this mean?
Because we release out of the whole pentapeptide library, and we release on the USM library, so everyone can download from USM library online.
Oh, okay. So the peptide library can be used for maybe screening for other projects and other researchers have access to it. Is
Yeah.
that correct?
Yeah.
Oh, okay. And then from here we can see the two pentapeptides that you have found contains the W residues, which are tryptophan and also tyrosine and histidine, which all have rings, right? Have some kind of ring structure, aromatic rings.
Yeah.
So do you have any sort of hypothesis or explanation why you think that peptides with these aromatic side chains is attracted or can bind well to the target?
Yeah, I think to holding the stable combined with S109, the peptide stick, you can see that the tridobone ATA and this one, and so many hydrogen bonds interaction. I think both of them can hold the stable interaction between ligands and receptors.
Oh, okay. So does it mean that if you have other variations of this, you will also bind well? Perhaps inside your
I'm not
library,
sure.
there are
I cannot say if you
some
have
kind of
so many
combination,
rings and then you can bind in better. For my results, I think the peptide stick is a very important thing for the stability. Even in the tree peptide library, you can see it? No, this way. Sorry. Yeah.
Tree peptide library.
Yeah, you can see the peptide sticking,
Yeah.
peptide sticking, peptide
Yes, yes.
sticking.
Ah,
And
okay.
also the ODA, the peptide sticking is also important. So maybe
Oh.
it's the reason why only the ODA can degrade the only ODA can degrade the performance S109 fibers. Another one just stop EU prolongation, but ODA can disaggregate.
Okay, so OLA also has tryptophan.
No,
or
it's natural
aromatic side chain, is it? OLA.
It's a natural
OLA has
product. I'm not sure it's
natural product. I see.
yeah.
Okay, so it's not
It's
not a
a
protein
small
or
molecule,
peptide.
maybe.
But it has some kind of aromatic or ring structure in the
Yeah.
compound. Okay,
And in
so
this
that's
article,
good.
they mentioned the peptide staking is very important.
So okay. So that's why the screening were able to select similar kind of structures with some side chain with this kind of ring, right?
Yeah.
Oh, okay. Understand. So from the three peptides and then the pentapeptides, the free energy binding is better with the penta one. Did you compare, for example, between the tripeptide and pentapeptide?
Yeah,
Free energy
it's
bind?
better because you can
Oh, okay.
see only actually the tripeptide cannot achieve the negative A, and you can see that so many pentapeptide can achieve the negative A.
Oh, okay. So maybe other residues provide some kind of stability, even though the ring side chain is important for binding to the, what do you call that? Your protein is granulin B, but then supporting residues is also important to make it stable in the binding.
How to say? Because I don't screen all the whole pentapeptide library. I screen the pentapeptide library based on my tripeptides results. So they already include many rings.
Okay, I
So I just
understand.
put
Okay.
amino acid in I just put a tripeptide in the middle and the two sides.
Okay.
So yeah.
Okay. Yeah, it's interesting for me why this can happen. So in the terms of the biological activity of this protein, you mentioned earlier that is it related to, in the introduction, is it related to amyloid beta?
Yes.
So is
Most of
this
study.
related to Alzheimer's amyloid, or is it a different protein?
Because S100A9 is inflammatory factor. So you have the inflammatory first, and then you will and then you will cause the Alzheimer's disease. So many researchers think the S100A9 amylose is the attention core because they have the core first, and then the A beta fibers will cover or interaction with S100A9 cores, and then to induce the AD.
Okay. So perhaps in the future, by blocking or inhibiting this from forming, then you may inhibit or slow down the progression of AD.
Yeah, hopefully.
Okay. All right. Thank you.
Okay, thank you.
Okay, thank you, Dr. Eugene. Dr. Tashri, do you have anything to add?
Okay, I think I have a last question for Gentile
Okay.
Pan. Okay,
Yeah.
all right. Can you go back to your replicate? Your MD simulation you run for 3 times, right? 100 nanosecond 3 times for both or three of the two of the peptides, okay, and then one of the oli A.
Yeah.
Okay, all right. So can give me any reason why only 100 nanosecond and why you perform 3 times three replicate? Why 100 nanosecond and three replicate in this your study?
Yeah, usually researchers will choose the 100 nanoseconds MD simulation, and then there are other professor have a lecture in USM before, and he mentioned that if we only do once MD simulation, we maybe
Okay.
miss some introduction.
Alright.
So for that we need to do more time. Such as 3 times MD simulation, or we have the longer MD simulation, such as the 500 or something else.
Okay.
Yeah,
Alright.
to include all of the information.
Okay. Okay. Alright. And then so you mentioned that this Oli A actually is a compound, right? It's not the peptide, it's a compound. Oli A.
Yeah. It's a natural product
It's a
from
natural product.
olive oil.
Okay, alright. And then the significant interaction is pi-pi stacking, and also the hydrogen bond at residue I not remember. Okay, this pi-pi stacking between tryptophan 88, okay, and hydrogen bond arginine 85. Okay, because that's why I said before, if you is it possible for you to analyze the hydrophobic interaction, because only this compound, although it have its own structure and everything, but it can show the significance of the hydrophobic interaction between Oli A and also the protein. Okay, so in your study, I think you have the trajectory. Okay, you have the trajectory 100 nanosecond, and then three replicate and everything. I think why not you just use like PLIC profile ligand interaction, sorry, protein ligand interaction profile, which is online and also can be run by command in your terminal, and then you analyze the interaction lah. That's my suggestion lah from me. Okay, in order for us to know either between the tryptophan 88 in the protein with your peptide can also establish this type of interaction, hydrophobic interaction. Then it can be considered good also as well as Oli A. That's my suggestion. Jantau. Okay, you understand?
Okay.
Yeah, you can use, maybe you can use PLIC protein ligand interaction profiler. That can online and also can use by the command from terminal. Then you can get all the interaction that involved in hydrophobic, in the hydrogen bond. Okay, rather than you using the Amber lah. Okay, that you can get from the MMGBSA Amber Python script. Okay, that's my suggestion lah. All right.
Okay.
And then I think that's all, Prof. From me. I think others is okay, no problem.
Okay.
Thank you, Dr. Tashwik.
Okay, no problem. Okay.
Dr. Eugene, anything to add?
No more from me. Yeah, I think
All right.
no, that's all.
Okay, thank you. If that's all, thank you, Chintao. And I would like to invite Dr. Chung to chair the next session. Thank you.
Okay, thank you, Dr. Ong. Our next student, just hold on. Our next student will be Song Jia-yi, supervised by Dr. Ong, and our evaluator will be Associate Professor Dr. Koo Boon Yin and Dr. Lang It Sin. So since Dr. Koo, you are there already, right?
Yes.
Yeah, and Dr. Eric also, yes, still there, right?
Yes.
Yeah, okay. In that case, Song Jia-yi, you can start your presentation now.
Can I see
thank
the
you, Doctor.
student for a while? Make sure
Sorry?
not AI is presenting.
Oh, yeah.
Please turn on your video for a while.
Okay.
Then after that you can start your presentations. Yes. Yeah. You can turn off the video.
Just hold on first, Song Jia-yi. it seems like your voice is on and off. Don't really can hear a clear sentence from you. The line seems like broken and on, like I hear broken. No, it's still the same.
Uh, is that because of the video? If just because of the video, then maybe you can turn off.
Can you hear clearer now?
Yeah, it's much better. You can start now.
Okay. My research project is an illustration of mechanism of antiproliferative properties of Camelina communis against the cancer rhizolytes. And introduction, Camelina communis is an annual plant belonging to the Camelina family, and native to East and Southeast Asia. And it has many ingredients such as flavonoids, alkaloids, and phyllosaccharides. And it also has anti-inflammatory, antibacterial, and antioxidant and antiviral effects. Literature review. Other plants under Camelinaceae family and their extract have already shown a certain antiproliferative effect on certain cancer cells. And the Camelina communis only shown have the anti-inflammatory, antibacterial, and antioxidant effects. Based on this project's objective is to evaluate the antiproliferative effects on C. communis and explore its mechanism of regulating proliferation in cancer rhizolytes and non-cancer rhizolytes. And its specific objective is to prepare aqueous extract of C. communis and assess antiproliferative properties of C. communis aqueous extract against cancer rhizolytes using MTT assay, and identify cell death pathway induced by C. communis aqueous extract against the cancer rhizolytes. The research hypothesis is the aqueous extract of C. communis can inhibit the proliferation of cancer rhizolytes, and the aqueous extract of C. communis is not toxic to non-cancer rhizolytes. The antiproliferative effect of the aqueous extract of C. communis may be achieved through induced apoptosis or cell cycle arrest in cancer rhizolytes. The methodology is 48 grams of plant into pounder and use water boiling to do the extraction. And the obtain the extract will do the saline test and then do the MTT cytotoxicity assay. The drug concentration will 2 times zero dilution from 0.75 to 200 microgram per ml, and will detect after 24, 48, and 72 hours. And because the previous study has shown that 660 nanometer LED red line have a certain antiproliferative effect to the MCF7 and the MDMB231. This time we also detect after 24 hours plus LED 660 nanometer. Then we'll detect in cell death pathway, use flow cytometry to do cell cycle and apoptosis. This is this times after 24, 48, and 72 hours MTT results. First in MDMB231. At low concentration, the loss availability is 72 hours, but with the increased concentration, 24 hours got a loss availability. And 72 and 24 availability shows a decreased trend with the increased concentration, but 48 hours is slightly increased. This may due to the different cell cycle. And in MCF7, this results at the concentration also shows a decreased trend, but at low concentration, 48 hours availability is slightly higher than control. This may due to different cell cycle and the first times cytotoxic accumulated toxic. And in MCF7, 72 hours got a lost availability. And 48 hours is higher than the other two time point. In M7A, the changes is relatively small, and in 24 hours total loss availability, the toxic is higher than 24 and 72 hours. At low concentration, it shows a decreased trade, but with increased concentration, 48, 24 and 72 shows a decreased trade, but 24 hours slightly increase. This may due to the high concentration got a highest accumulated toxin. And this results in 24 hours. 24 hours MB231 got a lowest availability. And at the low concentration, M7A's toxic is very similar to MDMB231, but with a higher concentration. At the same concentration, Q reduce a certain amount of MDMB231. This actor also can kill a smaller amount of MCF10A and shows a low toxic to MCF10A when reduce a certain amount of 231. And the overall availability of MCF7 is slightly higher than MCF10A at a certain concentration. In 24 hours, this extra shows a higher toxic to MCF10A. And both MCF7 and the MDMB231's availability is higher than the MCF10A. So, and because MCF10A is a normal cells, this is 48 hours this time point may not meaningful. And in 24 hours at a low